gene cloning. So genetic cloning is a procedure that allows you to take a gene and make lots and lots and lots and lots of copies of that gene. So we're taking a gene of interest, that's the gene we want to make copies of, and we're producing lots of copies of it. This is done today in medicine, molecular biology, it's done for genetics, cell biology, all sorts of things. There's usually two goals. Goal one, produce large amounts of DNA that contain that gene. So you just want a lot of DNA so you can do experiments on it. The second one is that you actually don't want copies of that gene, you want copies of the protein that gene makes. This would be like if we were making insulin, or if we were making another type of product that cells use. We talked about pKa, that phenylcalorenic thing where people cannot process an amino acid. What if we could make the enzyme for them to process that amino acid and give it to them as an injection every day? Would that be amazing? It would change their life. With cloning, there's a couple different steps to it. The first step is that you are going to take a vector. This vector is the thing that's going to take that gene of interest and use it. A vector is usually going to be a plasmid or a viral vector. It's going to then take it and take it into some place where you can do something with it. So you put it inside the vector, you put the vector inside of a bacteria. The bacteria now is going to have your gene of interest, and it's going to make tons of copies. So the vector is what allows this to happen. The way that it kind of works is you'll take the vector's DNA, like you see here, and in picture A, you have a nice strand of DNA. In picture B, do you see where those red stripes are? That's where the DNA is going to be cut. We use something called a restriction enzyme to cut it. The restriction enzyme has a very small section of DNA. It binds that section, and it cuts the pieces. And one piece moves up, the other piece moves down. You then are going to pop the gene in there, kind of like, any of you ever have those pop bead bracelets when you were little or see them? Or for that matter, if you built something out of Legos and then took apart the middle of the tower and shoved something in between, that's how this is going to work. So you kind of break the DNA, stick in the piece you want, and then it kind of velcros back together. We call the parts that are overhanging their sticky ends, they stick in, and they're going to grab it, and it will now include that gene of interest. Does that make any sense at all? I'm not going to ask you to explain how this works on the test. I promise. This is another diagram showing how it works. But maybe you want to know, did I insert it right? Is my gene actually in there? Or maybe I want to know how large a piece of DNA is. Or maybe I want to know how many amino acids are in a protein approximately. One of the things that we use to separate macromolecules like DNA and proteins to be able to study them individually is called electrophoresis. Who did this in high school? Okay, a couple people. This is a really cool process. So you take a pipette and you inject a little bit of liquid into this substance that looks like pretty stiff jelly. There's little holes in it. And then you apply an electrical current to this gel. And some things are going to move down the gel. And the speed at which they move is related to how large they are. What do you think moves further? Things that are tiny or things that are large?
Things that are tiny.
Things that are tiny. It's also going to sort by charge, by mass. Things that are heavier are not going to move as far as things that are lighter. And it's going to give you different bands. We'll see in a moment why those bands are really useful. There you go. Do you see anything in this that looks the same? Any bands that look the same? Any columns, I guess, that look the same? So the columns go this way.
The first one.
The last one and the first one, they're usually going to be what we call our ladders. That gives us size fractions. So they're usually going to be similar.
The like 3 kind of in the middle where the 2 bars are almost attached to each other.
So that one and that one?
Yeah, and the one almost on the end.
Mm-hmm.
So the second one and like the 6th one.
So there's a couple of them that have very similar bands. That may mean that they contain the same thing, or that they're from the same person. How many of you have ever watched a crime show where they say, based on the DNA, you are in fact the criminal or something like that? You're the killer, you're the burglar, you're whatever. This is a way that they can actually quickly kind of test some of that. We actually do a lab in General Bio 2 where the students are given a set of DNA samples. One is that of a criminal, and they have 5 samples. And you match the criminal to the suspect that is actually the criminal by seeing which columns look the same under gel electrophoresis, because you can look at specific genes this way. It's one way that we test this without having to sequence the entire genome. It's pretty cool. This is also used for seeing, did I insert my gene in correctly? Because if you know it's supposed to have a band and one doesn't show up, you know that gene is missing. So let's say that there is supposed to be a band right here. Is there one in that column next to it? Towards the center? Nope, not at all. So you would know, oh, I didn't actually get the vector in that one. So why are we cloning genes and genomes? You want to determine the sequence sometimes. Maybe you want to see if somebody has a disease. Or maybe you want to see if you've actually discovered a new species. Who likes tigers? How many species of tigers are there? Children's first day of class, right? There were 9, we have 6 or 7 left. Subspecies, please. It's messy. Why do I want to make sure that we have tigers of the same group mating with each other? So that we keep the subspecies or species separate, right? You are correct that we don't want inbreeding, which is problematic if breeding through each other. But let's imagine for a moment you are in charge of a zoo and you have 2 different types of tigers. Would you want to make sure that you continue to have 2 different types of tigers? And then somebody says, hi, I'd like to donate a tiger to your zoo. You would want to genetically test it to find out what type, so to see where it belongs, wouldn't you? That seems really far-fetched. This happens with like animal parks and things sometimes. Sometimes we want to figure out how a gene functions. We do this all the time. How does this gene work? What does it do? What is it related to? Maybe you don't actually want to isolate a gene. You want to isolate a protein. And then sometimes we want to make gene products like vaccines, insulin. One of the very amazing ones we can now do is we can create the blood clotting factor for people with hemophilia. So we can give them something they can use when they get injured. And hemophilia, we know it's bad if they get a paper cut or a bruise. But have you ever thought how people with hemophilia would have surgery? That's really complicated and scary then. So being able to give them a clotting factor so that their blood clots is very important. We also do biotechnology with the idea of genetic engineering. With that, we're genetically engineering. We are going to be directly manipulating genes. We're going to be working on microorganisms, plants, or animals. And the idea of genetic engineering is the use of living organisms or the products of those living organisms for human benefit. So a very fast raise of hands. Who thinks genetic engineering then is bad? Who thinks it's good? So when we talk about gene therapy and gene diagnosis, this is when we're testing for human disease. We can test on a human. We can also test on a fetus or on a fertilized embryo. That's where it starts to get messy. So we can test on a fertilized embryo, where we have the egg and sperm outside the person, and we put them together in a petri dish, and they are now a fertilized egg, fertilized embryo. We can then test to see if they contain genetic diseases. You can then implant the ones that do not and destroy the ones that do. Does that seem ethically problematic for any of you? Because once it's an embryo, is it a person? It's a hard ethical question. That's something science can't do. We can't answer that question in science. As Christians, can we answer that question? And then what if we take it a step further? What if we genetically test a fetus? And they go, oh, we're really sorry, it has a genetic disease. I don't know. Would you want to know that your child is going to have a genetic disease? Probably. So you could prepare. Then the question becomes, maybe not for anybody in this room. Maybe not even anybody you know, but would they keep the baby now? Sometimes the answer is no. What do we think about that? It's complicated. Hopefully, the way we're going with this instead is doing gene therapy, where we can somehow take a vector, give it to you, maybe give you like a common cold that contains a gene of interest that could fix your disease. What if we could take somebody with a genetic disease and give them the correct genes? That would fix everything, wouldn't it? We're not there yet. We're not. This one's how we produce insulin. In E. coli, you hear E. coli and how many of you think food poisoning? You probably don't think, let me make insulin in the lab. But we do make insulin by taking the genetic code to make insulin, putting it into a vector, putting the vector into E. coli, and the E. coli then makes the proteins for insulin. It's amazing, and it revolutionized how we could treat people with diabetes. This one, less revolutionary, just kind of interesting. Do you see the mice that glow in the dark? Yeah, we've put genes in them that cause them to fluoresce. Probably from jellyfish. You can buy fish that glow in the dark that have genes from jellyfish in them. Because some jellyfish glow in the dark. Now this is a proof of concept study. This is not a, yay, what an amazing, groundbreaking, life-saving study. Mice glow in the dark. But the fact that we can make mice glow in the dark means that maybe we can take a gene and put it into a person to make their life better.
Could we put that same gene set in humans?
We haven't yet, but wouldn't it be amazing? Now, usually a lot of this work has to be done on an embryo.
It would just be interesting to see, like,
Would you like to glow in the dark, Fran?
it would just be useful sometimes.
Terrible.
It would be useful for no one. It would have been useful today when we had no power.
I mean, like, just like when everything starts and you use a flashlight, like, you don't have to worry about it because you're going to go in the dark.
But you glow in the dark all the time. It's not something you can turn on and off.
I know, but like, if you like charge it, like, don't you like have to charge it and like let it for a little while?
No,
No, it just,
it's based on bacteria.
well, that
Charge?
yeah, I remember like shirts.
It's not like that.
You know what I'm talking about with shirts though, like
Right, but it's not like that.
With, like when you turn off the lights, it's like
Not like that.
You would have some good perks though. You get your own room by yourself.
Everyone would think you've been exposed to radioactive material.
Everybody would think you're
What?
They would be wrong.
Blown in the dark.
Do what?
All right, so movie one glow in the dark. Good. What if we genetically modify plants? So we take a gene, we put it in, and suddenly we've genetically modified corn. We do this all the time. 88% of corn, 93% of soybeans, and 94% of cotton grown in the United States is genetically modified. And you don't know. Because we don't have to label things as genetically modified. We do label them as non-GMOs. We're going to take our vote right now. Who thinks genetically modified is good? Who thinks it is bad and you can only pick one, you can't be conflicted?
Oh man.
What?
I'm gonna go with good.
Good.
Good?
Yep, good.
We got
No, I don't think it's
5, 6, 7, 8, 9. Okay, so we're split about half and half. So we got some benefits. We can ease world hunger. With genetically modified food, we can feed more people on less land. Is that a good thing?
Yeah, just but we don't do it.
Also, we're going to develop crops that we can grow in bad soil, in drought areas, and areas of saltwater intrusion, and we can grow things that are going to be less likely to spoil.
But why don't we do this like with California a couple of years ago is having the drought epidemic. That was last summer, right?
Yeah,
You ever have-
still in drought.
Yeah, and what is it? Why? And they were talking about like how all their crops were. So like, why don't if we have?
Because these aren't what we have there. And because these aren't grapes and lettuce.
Wait, it only works for grapes and lettuce?
No, that's what California mainly grows in those areas.
Oh.
It's not the same crop.
So you can't do.
We can't do it with every crop so far, no.
Well.
Lettuce is mostly water. We can't have a drought-resistant lettuce. We have drought-resistant other things. We can reduce the strain on non-renewable resources by having crops that need less water and less fertilizer. This sounds fabulous, doesn't it? More good. We can improve our crop quality. We can use less pesticides by having disease-resistant crops. We can have crops that can grow in areas where there's frost on the ground. We can have crops that can live through a flood. And there's groups of people in Africa that don't eat anything that's the color orange. What do you know about eating food that is orange? Why do you need to do it? Tim. It's good for your eyesight. What do you think happens to people who don't eat anything orange? They become blind and develop cataracts. We have discovered that by enriching rice with the vitamins that they're missing from the orange food, we can solve their vision problems.
So you don't eat orange?
You took multivitamins as a kid. We're
Yeah, but like I'm saying, like
If you didn't take multivitamins and you didn't eat things that were orange and contained vitamin C, A, and beta carotene, eventually you would develop vision problems. Especially if this happened while you were a child. Or what? There.
Well, how much oranges would you have to eat?
Not much. But you also probably took a Flintstones vitamin every day when you were a little kid.
I never took vitamins, but I ate
Carrots.
so.
And you probably eat cereal and things that are fortified with vitamins.
Yeah.
Our diets are very fortified in vitamins. This is not true in other places in the world. Look! We can make things resistant to disease. These transgenic plums, they look beautiful. They are resistant to the plum pox virus, which is amazing. Flip side, some risks. What if insects actually can overcome the resistance in genetically modified food, and then we actually have to use more pesticides? Happens.
How?
Not often yet, but it will happen. We have discovered that also, sometimes these pesticide traits actually can keep pollinators away too. There's is a bigger problem. We haven't quite figured out that it's a huge problem yet, but it might happen. And what if these weeds, or these crops, let's say a corn crop, somehow magically cross-pollinates a weed? Unfortunately, in plants, this is really common. Things will cross-pollinate that shouldn't. And the traits are now passed into the weeds. And the weeds are herbicide and pest resistant. You've now created a weed problem on your farm. Anyone have a food allergy? Nuts?
Some nuts and fruit.
What if we took some genes from peanuts or tree nuts and put them in wheat? Would that be a problem for you?
And wheat?
Yeah.
Yeah.
What if it made the wheat better? Would it still be a problem for you?
Yeah.
Potentially, right?
Yeah.
Exactly. And we don't label what we genetically modify. Right now, we're not genetically modifying anything with known allergens. But what if that happens? There's that fear there. And what if we modify things in a way where they're no longer the same? And then, well, how comfortable are y'all playing God? Because this is running kind of close to that, right?
Yeah.
Oh, and then there's the fact that, of course, GM foods are regulated, not regulated. Having said that, there's no studies that prove that they're any worse or any better unless they contain additional nutrients than non-genetically modified food. So letting us feed more people, letting us introduce additional nutrition, they do pose potential risks and potential problems. And they don't appear to be different. So every time you see science now says genetically modified foods are not as good for you as normal foods, there's no study to back it up. We don't see a difference. Your body doesn't see a difference between corn that's not genetically modified and corn that is genetically modified.
I mean, it just sounds like there's more good to outweigh the risks.
And potentially I also structured it that way. Part of it is that a number of the risks are coming from science studies that aren't really science studies. And that's part of the problem. Because the funding that comes from this work is usually related to companies like Monsanto that produce a lot of genetically modified crops. So there's that problem too, is that we don't always know enough. Do I eat genetically modified things? Sure. I don't really know how to avoid it because lots of things are genetically modified now. Am I constantly worried about it? No. Do I know people that are? Yeah. And I think it's a choice you get to make. Kind of our last big thing is the idea of cloning animals. Reproductive cloning is what we're dealing with, and in this, what happens, get caught up, with reproductive cloning is that you are going to clone a multicellular organism. But it's a lot different than cloning a bacteria. You can't just make a copy of it. You are going to, however, take the DNA from a nucleus of the cell, put it into an egg, and then let it grow and develop into an adult eventually. So the example over there in cartoon form is of Dolly the sheep. Who has heard of Dolly? Dolly is our first big clone mammal, right? Dolly, they took a nucleus from the one that gave us Dolly, from a mammary cell. They put it into an egg from another sheep, and they allowed it to grow. They implanted it in a third sheep, and then Dolly, the baby sheep, was born. So she has a nucleus of one, the egg of another, and then a third carried her to term. Complicated, right? What do you all know about Dolly? You don't know when it happened?
She died.
She did die eventually. It happened in 1996. She was born. Dolly was almost genetically identical to the one that gave the nucleus. There was a slight difference due to mitochondrial DNA inside of the egg. Dolly did grow like a normal sheep. She lived and died like a normal sheep. And since then, we have cloned sheep, cows, mice, goats, pigs, and cats. The cat was the first cloned cat in 2002. Someone wanted to clone their cat before it died. They took a sample and sent it off to a lab in South Korea to get a kitten back that was the same as their cat. They named it carbon copy. They called it CC for short. It was not quite identical in behavior, though. Right?
Or just buy a cat and be like.
But it wasn't the same cat, Ethan!
Yeah, that's what I'm saying. Like, if she said.
Yes, and it would have been much cheaper.
Oh,
No,
you mean it's the
I'm saying, like, yeah.
Oh, like the company just sat around.
Yeah, like.
Somebody wanted it done.
Yeah, no.
You can do it right now. You can clone your animals. I'm not suggesting it. You can also get stuffed replicas of them. Yeah. Have we cloned people yet? North Korea says they have. No one believes them. But we could clone humans, right?
It's the same thing, right?
Can you imagine? You need a heart transplant. Take your cloned human out of your closet. Kill them. Take their heart. It's yours. Sci-fi movies are based on this. The clones take over. How many of you want to clone for spare body parts? This is part of like the idea where people are like, we can get them for spare body parts. How many? We can, moving on, use DNA fingerprinting in determining crimes and determining things about who did what. And then finally, the last, I think perhaps best part of the biotech stuff right now is biofuels. The first one I don't think is useful. Ethanol from corn, and we could use corn for better things like feeding people as opposed to making fuel. The new plans for biofuels are coming from algae, yeast, and cyanobacteria. What if we could use them to create all of the fuel we need for all of our cars? This would actually decrease carbon dioxide pollution. It would actually increase carbon dioxide sequestration where it comes out of the atmosphere as these organisms do their daily things. It's renewable. It would probably produce oxygen or water as a byproduct. There's a ton of ongoing research on this. Plus, think how cheap fuel would be if it actually came from yeast.
Doesn't like all of Brazil's like cars with off of sugarcane ethanol? Because they can like mass produce sugarcane that way.
Not all of them, but yeah, they're doing a lot of that. A lot of places are. There are plants that can grow in salt marshes that we can take the protein from and use as a protein source. And the ethanol that's pressed from them is larger quantity and higher quality than we get from corn, but we're not doing it. So we could someday have fuel that's not oil and not ethanol from corn powering the car. Or, for that matter, waste oil from McDonald's, which you can get the kit to potentially change your car to run off of waste food oil. But then your car is going to smell like McDonald's. That is the end of